a warm welcome to the third MAC-ID conference. Acute febrile illness is any clinician's dilemma due to the difficulty in approaching a diagnosis. An accurate diagnosis is important for the selection of appropriate treatment for the acute febrile illness. We have today Dr. David Richard Bell, who specializes in innovation in global health and health technology development, translation, and introduction. He will be speaking to us on diagnostics of acute fever, technology innovation meets reality. Welcome to you, sir.
Okay, thank you. And thanks first very much for inviting me to Manipal and for the weather also, which is excellent. So I wanted to talk briefly today about acute fever, thanks to the introduction, which has become more prominent with malaria, increasing use of malaria diagnostics, but is not new, it's just being uncovered and more prominent in front of us. And some of the issues with the approach and the lack of progress, perhaps in the last 20 years on this, and where we might sort of address these more effectively in the future. So, the introduction of malaria rapid tests about 20 years ago, which sort of brought a lot, particularly throughout the malaria endemic world, brought to prominence non-malarial fever through essentially removing the question of malaria from this. So previously people in a lot of countries, particularly sub-Saharan Africa, treated fever as malaria. The introduction of rapid tests, most of them were negative. A lot of countries initially thought they weren't working, but they were. It's just that they'd been mistreating fever for a long time, and most fever, in fact, was due to other causes. And the top graph there shows the introduction in really the first large-scale community-level use of RDTs in Africa, in southern Zambia, where they'd had reported about 10,000 cases every quarter of malaria. And with the various interventions like ACT, indoor spraying, very little changed. Introduction of rapid tests, and they suddenly dropped down to about 30 per quarter. So it wasn't that malaria had suddenly dramatically disappeared, it was that they had always been mistreating other causes of fever as malaria. So this is helpful, but it also raises a big dilemma for the health system. Malaria was no longer one of their major, top 10 diseases, but something else was, and they didn't know what it was. And the lower graph is from Senegal and shows a very similar issue. So they introduced rapid tests and better microscopy, which is the red line there. And you can see that the use of ACTs drops dramatically with this increase and eventually matches the positive rate for malaria diagnostics, but there is a big gap there. Those people still have fever, but it's very unclear what they have. So these people are still sick. And sick with what? So this is some example on the left from Tanzania, on the right from Laos, of trying to find out what these people have. And I mean, the story is that there is, apart from malaria, a huge range of viruses, bacteria, some parasites. Most of the viral are probably predominant. Most of these are self-limiting infections that if the person never gets treated, they will get better. But a smaller number of these are potentially very severe, and some, such as rickettsial infections, typhus and so on, are really only becoming clear now with a more effective treatment of management of malaria. And these vary a lot from place to place, but there are some, the underlying rates of probably about 10 to 15% rickettsial type infections, predominance of pneumonia, etc., persist across geographies. So the question is what we can do with these, and you clearly can't have a diagnostic for each of these pathogens. So thinking about someone comes with an acute fever They may have a malaria infection, and malaria infection, not all malaria infections are febrile, so that's another issue. That's diagnosable and treatable. They may have other diagnosable pathogens, but many of these are self-limiting, so we can spend money trying to diagnose them, but that doesn't necessarily help public health. Many of these are probably not really diagnosable. They're viral. We could sort of dive in and try to find them, but it's hard to prove that they're even the pathogen in question, even if you isolate them. And they don't matter in terms of long-term health. And a very small number of these can produce very severe disease or mortality. So this is, when you add these up, this is still one of the major causes. Acute fever is probably implicated in the major causes of childhood mortality, preventable mortality, infant mortality, and a lot of adult mortality. So although many of these don't matter, it's vital to work out, to identify those that are severe and that do. Looking at this also offers other benefits. So this is an example from Myanmar where and it's a malaria control program across a lot of clinics at community health level, and they had a rapid decline in malaria, but also in attendance rates at the clinic, because when people come with malaria, they have a fever, they have a malaria test that's negative, they're told to go home. It's not a big incentive to come to the clinic again. So they introduced a raft of other interventions, or including sort of diagnostics, etc., but also other clinic activities to address the needs of those patients. And you see an increase in testing rates again. So in 3 of the 4 clinics, there's a big increase again in the number of people coming, and also in so this benefits malaria. You're getting a lot more malaria screening, but it also benefits health in general because you're getting a lot more attendance at clinics. So the clinics need to address fever in order to be credible in the eyes of the community. So this has been apparent for quite a long time. I decided to go through some issues around technology development, why we may not have got far. So this is human population distribution, which is very much skewed towards Asia and Africa, so it's redrawing country boundaries on the basis of population. This is scientific papers published, which is a proxy of where product development, etc., is happening for diagnostics, and it's nearly all in Europe and North America. There's an increasing amount in India and China, but it's very skewed away from where the large part of the human population is, and it's extremely skewed away from where the problem is. So we have a lot of people excited about acute fever and diagnostics and developing something for this. They're not in the place, and they're not that closely connected often to the communities that have this problem, that are dying from this, and that understand the real needs of the people and what's happening at a clinic level. So, I mean, India is an interesting case here because it has a significant amount of research going on, but it also has significant mortality and a significant population. So it's, in a way, it's in the middle, and it's very well placed perhaps to be one of the leading sites in addressing technology development here. So if we think that health is a fundamental human right, we need to address this. And this probably means either, so I've got small buttons here and big fingers. So we need to address this maldistribution of where funding goes and where product development takes place, either through very good communication or through shifting where funding and where things happen. And much more strongly engage within these countries, I think. So somewhere like India and Manipal should be really one of the centres of addressing this. So switching quickly to malaria as an example of this. So this is even more skewed if you look at where malaria deaths are occurring. Apart from RDTs, and it's now a quarter of a century since we had rapid tests for malaria, there's been a huge effort, and this is just an example, a few examples of the technologies that have been specifically targeted towards malaria in the last 20 years or so. And a few of these will be useful, but we're still using 150-year-old microscopes and 25-year-old rapid tests. So clearly this technology development, which is for malaria, one of the major causes of fever, is somewhere misplaced. And the people that are developing these with great intentions are not really connected and not understanding the problem. So we need to do this if we're going to progress, and particularly even more so for the other causes of acute fever. So thinking about the context that fever occurs in, we can develop diagnostics for it, but we're trying to stop people dying. And people die because of the pathogen, but they also die very much so because of underlying diseases, poor nutrition, environmental, etc., access to health services, incompetence in delivery. And technology is an addition to these, but because we have a huge problem with fever doesn't mean we have to develop new technologies. It may well be more effective to address these other issues or to develop technologies which will facilitate that. So understanding whether tests work in the context of a clinic, there's diagnostic test is one aspect of finding out what's wrong with the patient. So just walking through what happens when a patient with acute fever comes to a clinic. And they come because they feel unwell, they have a fever, they may think they have malaria or something else, but they're seeking in the end a diagnosis and management. The vast majority of these will be quite well. There'll be some routine management they can have, they'll be discharged home. And most of these people, they may not need specific treatment, they will get better anyway, and they just need to be confirmed that they're not going to get very sick. A very small number of these will have evidence of severity on initial triage and will need much more deep investigation and perhaps referral if we're going to mitigate the sequelae of severe illness. So the function, the primary goal of managing acute fever is really to identify these people and to manage this rather than to identify the individual pathogens which may be contributing to the fever. So there are a number of ways that we can do this, and they all have various costs. So at the moment, the IMCI protocol for integrated management of childhood illness is the WHO's attempt as a paper-based algorithm essentially to try to identify fever and effectively manage it. And it's extremely cheap, but it's extremely non-specific and fairly poorly implemented, but it's probably the cheapest intervention we have. We can, particularly with the increasing amount of data now, and with increasing use of artificial intelligence, etc., there's a potential to have sort of dynamic clinical algorithms where we can essentially make these algorithms much more fluid, responding to the environment. And this is, potentially we may manage to identify most cases that need further intervention in this way, again at a fairly low cost. Next we can look for, we can think about developing diagnostic technologies which will identify bacterial and viral, or which will identify markers of those who will become severe. This is adding cost, but it's adding specificity. And lastly, we can try to develop diagnostics for all these pathogens, and this is the most costly approach. And somewhere in the middle here is probably where, for most low-income countries, we need to be. So how do we do that? The idea of dynamic clinical algorithms is gathering increasing interest. A patient comes to a clinic, sits in front of a health worker. We can take the context of the patient there and a diagnostic test at that time, or we can take into account in that decision-making process nearby outbreaks such as dengue, etc., seasonality, geographical variation within a country, and then feed into that specific diagnostic test which might be recommended by the algorithm. And we can do this potentially without the health worker necessarily knowing that there's an outbreak, if we can use data effectively and inform this through a tablet, etc. So potentially without a lot more training, we can build on the sort of IMCI type protocols, make them relevant to the place, relevant to the clinic, and relevant to the particular patient. And there are some efforts to do this. This is Health Cube from actually based in Bangalore. And they've developed essentially a box which is just an integrating box. So it has cameras in it that will read rapid tests. You can plug in a blood pressure, cuff, a pulse oximeter, ECG module, etc., hemoglobin, uric acid. But it doesn't just give results. The idea is then it will integrate into the EMR, the electronic medical record system. It will take into account other aspects of the patient, and it will guide the clinician in diagnosis. So you can potentially take a quite low-level health worker and have them sort of fairly cheaply making the sort of decisions that you would make perhaps at a physician level at a hospital. So this is realistic. It's not expensive. It's a way of using technology to much better integrate the diagnostics and the data that we already have for decision making. We can also improve its vital signs measurement. This is an example from University of California, San Diego, that is under development, where you can have a simple device, and there are others like this, a single device that will measure temperature, pulse oximeter, ECG, etc. So you can take very accurate vital signs and you can feed these directly into a digital algorithm. And potentially you can also then be taking photos using AI, image recognition, etc., to make other diagnoses. So we can use technology without specifically diagnosing to greatly improve the quality of triage and identify those who need more care. At the next level, there's some increasing interest in biomarkers for identifying severity, or those who will become severe. STREM1 is a really interesting one. With it is Roche, for instance, is developing a test for sort of rich ward applications on this at the moment. These are all in the cascade that leads to capillary leakage and the acute phase or activation. And if we can identify these changes very early on in terms of biomarkers before they actually start to have an effect on the body, we can potentially identify very early those people with acute fever who are going to go on to have decompensation and potentially mortality or long-term sequelae. So there are a few of these, such as STREM1, angiopoietin-2, etc., that are in sufficient concentrations at thresholds of interest that they could potentially be put on a rapid test. Further, there's some evidence that even just measuring better the underlying illness, particularly hemoglobin and weight for height, will almost be as good as these in identifying who is going to get sick. So a kid with diarrhea will usually get better, but if they have a low hemoglobin, if they are low weight for height, they will usually not, and they warrant referral. So much better use of these sorts of biomarkers and existing tests. So further distinguishing bacterial from viral illness, which has implications for AMR as well. Excuse me a moment. So if we can identify infections that warrant antibiotic or antimicrobial use, we can go some part of the way to narrowing down who we need to concentrate on, who will benefit from treatment. If we can identify classes of antibiotic use, such as tetracycline sensitive versus penicillin sensitive, we can go further. And there are a number of products close to market or touching the market that can potentially do this. So C-reactive protein is fairly non-specific, but is being used in a number of studies now and has reasonable definition in terms of distinguishing bacteria from viral. Procalcitonin perhaps not so. The February DX has a test out now in fairly high-end applications, but the underlying technology is quite cheap, which will more accurately distinguish bacteria from viral through addition of MXA detection. MEMED has another one which is relatively expensive. So there are a number in the pipeline or close to market that will distinguish bacteria from viral. They are too expensive now for use in most clinics, but the underlying technology is there and they need to be made cheaper. So we will still be left with some diseases which we have to diagnose and identify the individual pathogens. So typhoid is an example, group A strep. It may not kill the patient immediately, but it has big implications long term. Melioidosis, obviously, you need to identify. Dengue, Zika virus, some of these will not be identifiable through just measuring severity. So we are still going to need a number of specific diagnostic tests, but we can be very selective probably in which pathogens we need to put on these. And we'll need new technologies to do this. This is an example of devices in trials in a few places at the moment for identifying group A strep or identifying bacterial infection in the throat and distinguishing that from viral. So essentially it's an image recognition system, a camera with some AI, and it'll diagnose at essentially zero cost as it's once you have the device, there's no consumables, but with more accuracy than a rapid test. So there are a number of technology is changing for these very few pathogens that we probably have to identify. There are paths to do that. So just reflecting on how to get these to market, and getting a sensitive test does not mean it's going to work at point of care. So in product development, we need to take into account very much the use of the test in the context that I showed earlier. So a lot of tests fail, poor transport, poor preparation, poor interpretation, poor use of results, and this will lead to poor outcomes and poor reputation for the tests that won't be used. So all these costs, so the introduction of any diagnostic, they often look cheap, but in a programmatic context it's extremely expensive. So again, this emphasises really the fact that we can afford very few diagnostics at this level. We've got to be really selective in the pathogens that we aim for so that they actually make a difference. And probably we need to then concentrate a lot more on these tests which are going to identify who needs further investigation and further referral, rather than trying to identify a range of pathogens individually. So just reflecting on what these assays need to be: simple, affordable, etc. They need to be needed, so they need to be developed in the context of the communities and the health systems that are going to use them, which is why I think we failed to a large extent in the past, in 20 years, to make much progress. They need to enable changes in management, so knowing the pathogen is not useful if you can't do anything about it, and they need to impact on outcomes. They need to be simple enough to be accessible and have minimum consumables. So just to summarise overall, acute fever, it's underlying the cause of most preventable deaths at this level. We shouldn't be talking about non-malarial fever, we should be talking about acute fever as a syndrome. And thinking about then, malaria is one of the tests that we use on the path to identifying who actually needs something done. There are very few technologies that have reached point of care at this level, partly because I think the developers are not understanding the true needs. We need a much more holistic approach to this, to think out not identifying pathogens and treating them, but thinking out why the patient has come and what actual interventions are going to make a difference in the long term to a patient. The key to this is improving the specificity of triage, so understanding which patients will benefit from further intervention and which patients are likely to die and need referral. So technology will be part of this, and in terms of diagnostics themselves, we probably need new platforms to make much impression. We're close to where we can get with the current platforms. So, thank you.
Thank you very much for sharing your vast knowledge on innovation, especially in the identification of biomarkers, severity of illness, as well as identification of etiological agents. The talk is open for discussions. We have 5 more minutes and we can take a few questions.
What about multiplexing of diagnoses for multiplexing?
Yeah, so, and it'll be nice, yeah, and people thought for a long time, say, on a lateral flow to put multiple lines. And, you can imagine having dengue, chikungunya, and malaria, and a few others. And it seems really simple and obvious, but it's actually, one of the problems is that there's geographical variation and there's seasonal variation. And so you end up with a very small market where the test makes sense. And if you're making a lateral flow or any diagnostic terminology for a very small market, it becomes very expensive. Malaria tests are cheap because they're made in the hundreds of millions. And you then have a problem if you have one test with multiple pathogens, that if the dengue test is not quite as good as the standalone dengue test, do you pay for the multiplex test and risk missing some dengue cases, or do you still buy the more sensitive standalone dengue test? So it seems an obvious thing to do, but on the current technology, it's hard to see how you can do it and make a really marketable, commercially viable test. I think eventually the time will come, but it's probably going to be on other technologies. And there are some emerging technologies in the immunoassay field that might be able to do this. They're reasonably sensitive and potentially very high multiplex and cheap, and they're not based on paper. So they're potentially easier to manufacture, but they're not there yet.
Which of these technologies are actually being used? So many of them that you shared with us today?
Yeah, so, CRP is being used to distinguish bacteria from viral, but there's a lot of controversy on that. Perhaps it's not as specific as perhaps it should be. The integrating technology that you saw, the HealthCube device, is in use. It's not a test in itself, it's just integrating it's integrating information from current tests and using that so that can be used much more efficiently. Roche is developing an S-TREM1 test, which has potential, say, in emergency departments and so on, in high-resource settings, and it will be a relatively expensive test. But that's something that you could put on a lateral flow and make quite cheap. The problem is, with all of these, is that you can develop a test somewhere like Manipal, in your biotechnology department, but making it manufacture of high enough quality and cheap enough that it then makes public health sense in Myanmar or southern India or Malawi is a huge step, and you have to get the stability, you have to get all these other things in there. Plus you then have to have a health system who wants to buy it and do something with those results. So a huge step. So there's not a lot of sort of triage going on at the moment, but it is probably where things will move because it is a much more practical approach than trying to have 20 different pathogen tests or trying to have very high multiplex on current technologies.
If there are no more questions, we can close the session. Thank you so much, Dr. David Berger.